The patient received the drug that contains harmless virus particles that were produced in the laboratory. The virus particles identified only infected cells because of antibodies attached to them that bind to specific receptors on cancer cells. This is how they merge, with the virus inserting its contents into the cell. The HIV virus binds with healthy cells and injects RNA into them. In a reverse process, the RNA becomes DNA and enters the cell's nucleus along with the integrase, an enzyme that merges the viral DNA with the cell's DNA. When it comes to cancer cells, we produce lentivirus particles in the laboratory, which allow the same process to occur. In a normal process, the cell identifies fractions in its DNA and corrects them through a natural healing mechanism. The scientific discovery and the breakthrough of GOMMORA enabled a peptide, a tiny protein segment, to bind to the integrase in a strategic location. The peptide binds where another protein should have connected, a restrictive one, which goal is to prevent further penetration of viral DNA fragments. But now, the peptide prevents the cell from stopping this process and forces it to merge additional viral DNA fragments into its nucleus. Most of the existing drugs try to delay the activity of the integrase from integrating diseased DNA into the nucleus of the cell. GOMMORA does the opposite. It increases this activity until the cell cannot withstand the load, fails to correct itself, realizes that there is a problem, and destroys itself. The virus can identify and connect only to infected cells, therefore they are the only cells that die, leaving healthy cells unscathed, thus operating without side effects to the patient.